Today we'll talk about the Hershey and Chase transduction experiment. This Hershey and Chase transduction experiment is another evidence which suggests that it is the DNA which acts as a genetic material in organisms. Actually, two experiments were being performed as evidence which says that it is the DNA which acts as a genetic material. One was transformation experiment by Griffith that we have studied, and then another is transduction experiment by Hershey and Chase. This is also known as bacteriophage experiment by Hershey and Chase. This Hershey and Chase, they named their experiment as blender experiment. Blender experiment, also known as blending experiment, because in this experiment, these scientists Hershey and Chase, they blended this E. coli and bacteriophage, T2 bacteriophage together in a agarose medium. So they used the term blender experiment. Actually, blend means to mix. In this experiment, they have mixed together E. coli and T2 bacteriophage into an agarose medium. These Hershey and Chase, they experimented. What their experimental stuff was T2 bacteriophage. They experimented with T2 bacteriophage. Hershey and Chase, they experimented with T2 bacteriophage in 1972. They performed this transduction experiment with T2 bacteriophage. Actually, this T2 bacteriophage, it is a virus that infects E. coli. So sometimes this T2 bacteriophage, it is also called as coliphage. As you have zoophages, viruses that infect animals, phytophages, viruses that infect a plant. Same is the case, we have coliphages. For example, T2 bacteriophage. This T2 bacteriophage, it's called as coliphage because this virus infects Escherichia coli. If we'll structure the structure of this T2 bacteriophage, it has two important components. One is DNA and another is protein coat. So this T2 bacteriophage, it is having this is called as protein coat, what is called as capsid, and inside this protein coat it is having a genetic material, viral genome, DNA as a genetic material, and this is called as collar, and this is called as tail. They're called as tail fibers. This is the typical structure of T2 bacteriophage. By virtue of these tail fibers, this phage, T2 phage, this phage gets attached to the E. coli and transfers its genetic material. For example, this is E. coli. Now, what was their experimental stuff was bacterio T2 phage, which infects Escherichia coli, E. coli. The objective of their experiment was to prove that it is the DNA of the phage that enters into the E. coli, while as this protein coat outside, it is made up of capsomers. This protein coat, it remains outside. So they want to prove it that the DNA, what they want to prove, that DNA enters this DNA, they want to prove DNA enters the bacterial cell E. coli cell, and protein coat outside, and protein coat remains outside. So they wanted to confirm this, that it is the DNA which enters into the E. coli and the protein coat remains outside. You know that this phage has two important components. One is DNA, it is phage viral genome, and another is proteins. This protein, it is in the capsid, and this DNA, it is simply a viral genome. The underlying point in this experiment was that it is the phosphorus which is present in the DNA, and phosphorus present in DNA. You know that it is a phosphorus which is present in DNA, and this protein, it contains what? It contains sulfur. It contains, because we have many sulfur containing amino acids in a protein. For example, we have cysteine, it is a sulfur containing amino acid. We have methionine, it is a sulfur containing amino acid. So they use two radioactive tracers. One radioactive tracer was phosphorus 32. It was used to make DNA radioactive, to make the DNA of phage, T2 phage radioactive, and sulfur 35 radioactive trace, sulfur 35 radioactive tracer was used to make protein coat radioactive, protein coat radioactive. So what they did, they used two radioactive tracers for their experiment. One was phosphorus 32 to make the DNA of the phage radioactive, and then another radioactive tracer, that is sulfur 35, to make the protein coat or capsid radioactive. In their experiment, they made two cultures. Culture first, culture A and culture B. In the culture A, what they did, this is culture A, they took the phages, this is T2 phage, this is T2 phage, and they made their T2 phage, and they made their DNA radioactive by means of phosphorus 32. In culture A, they took the T2 phage and their DNA was made radioactive by means of phosphorus 32. And the culture B, they also took T2 phage, and here in the T2 phage, these are T2 phage, here protein coat is made radioactive. Protein coat, that means capsid is made radioactive by means of sulfur 35. So in this culture we have got T2 phages where DNA is the radioactive, and in this culture B we have phages where protein coat or capsid is the radioactive. Now these two cultures were used independently to infect the two E. coli cultures separately. Now this culture A, it was used to infect the E. coli culture, and this culture B, it was used to infect another E. coli culture separately. For example, we have this E. coli culture, E. coli culture, E. coli culture, and this T2 phage where DNA is radioactive, it was used to infect this E. coli, this E. coli culture. Same is the case where this culture B, the T2 phages in the culture B, they were used to infect another culture of E. coli. These are E. coli. These are E. coli. So now they followed three important steps in their experiment. This was the basic idea about their experiment, that they made the phosphorus of the phage radioactive by means of, they made the DNA of the phage radioactive by means of phosphorus 32, and they made the protein coat of the phage radioactive by means of sulfur 35, and then separately this the phage having the radioactive DNA was used to infect the E. coli culture separately, and the phage having the protein coat radioactive with sulfur 35, that was used to infect separately another E. coli culture. And three steps were followed. One was infection. One was infection. Three steps were followed. One was infection, second is blending, and third is centrifugation. So first was infection, second was blending, and third was centrifugation. So infection simply means that where E. coli was infected with the two phages. One phage is having the radioactive DNA, another phage is having the protein coat radioactive. They were used to infect separately. They were used to infect the Escherichia coli. Then in the blending, now you have got E. coli and phages are together in a medium, and then these are taken in a blender so as to separate the bacterial cells from the phage particles. And third case, in the centrifugation, in the third step, centrifugation, in the centrifuge tube, the heavier these bacterial cells, they were present in the heavier bacterial cells, they were present in the bottom, that is in the pellet, and the empty protein coat, they were present in the supernatant. So during infection, these phages were used to infect the bacterial cell, and these phages entered their viral genome into the bacterial cells. In the blending, then these phages were separated from their bacterial cells. Then in the centrifugation, it was in the centrifuge tube, it was found that the heavier bacterial cells, they settled at the bottom, that is in the pellet, and the lighter protein coat, empty protein coat, that remained in the supernatant. That we'll explain like this. So we have Well, how they performed this experiment? This is bacteriophage. This is bacteriophage. We will take culture A. This is bacteriophage where DNA is radioactive. So here this is T2 phage. This is T2 phage, and we have radioactive DNA. Radioactive DNA. That means this DNA contains what? It contains phosphorus 32. And this T2 phage was used to infect what? E. coli cells. This is E. coli cell. This is E. coli cell. E. coli cell. This phage is used to infect the E. coli cell. This is called as infection. Called as infection. Obviously this, now you can see that I have shown small lines on this DNA. It means that this is a radioactive DNA. This is a radioactive DNA. These small lines indicate that it is the radioactive DNA. We have made the phage DNA radioactive by means of phosphorus 32. Then obviously this DNA will enter into the E. coli. So it would be like this. It would be like this. So we have what? This is phage DNA. So this is phage DNA, yeah, viral DNA, viral genome, viral DNA. This is the genome of E. coli, viral genome. This is called as when the protein coat is empty, it is not having DNA in it, capsid is not having that DNA in it, that is called as ghost. What is ghost? Ghost is empty. Ghost is empty protein coat. The empty protein coat is known as ghost. So this was the infection. Then we have blending. We have blending. That means these, in a blender, the bacterial cells were shaken so as to break the physical contact between the viruses and the E. coli cells. So we have, these are E. coli cells where we find viral genome, viral genome, viral genome, plus we have what? We have ghost here, empty protein coat, empty viral protein coat, empty viral protein coat. So this is infected bacterial cell, infected bacterial cell, infected E. coli cell. That means it is the E. coli in which viral genome has entered. That's called as infected E. coli. And these are called as empty viral, empty protein coats of virus. Empty protein coats of virus. Those are called as ghosts. Those are called as ghosts. So these infected E. coli are heavier, and they will settle. These infected E. coli, infected bacterial cells are heavier. They will settle down in the bottom, in the centrifuge tube. That is, they will form the pellet. And these empty viral protein coats, they remain in the supernatant pot. So we have then centrifugation. We have centrifugation in the centrifuge tube. In the centrifuge tube, the heavier bacterial cells, the infected bacterial cells, they will settle down in the bottom, what is in the form of pellet, and the lighter empty viral protein coats, they will remain in the supernatant, and all the radioactivity will be associated with the pellet. So we would have like this in a centrifuge tube. This is centrifuge tube. We will find this is centrifuge tube. We will find that radioactivity is here. This is radioactivity in the pellet. So radioactivity in pellet, and these are empty, or we can say that ghosts in supernatant. Ghosts in supernatant. This we know that in the pellet what would be present? Bacterial cells. Infected bacterial cells would be present in the pellet. These are infected bacterial cells. Infected bacterial cells. Infected bacterial cells are present in the pellet. So in a centrifuge tube, when we have taken them in a centrifuge tube, we know that the heavier, denser particles, they will settle in the bottom, and the lighter particles, they will be in the supernatant. This part is called as supernatant part, and this part is called as heavier part. This part is called as a pellet part, and this part is called as supernatant. And the pellet, we find heavier particles. That means we find bacterial cells. And in the supernatant part, we find empty viral protein coats, what are called as ghosts. We know that this infected bacterial cells are present in the pellet. That and radioactivity, we see radioactivity in the pellet. It means that this viral DNA has entered into the bacterial cells, and we find radioactivity in the pellet. Why we find radioactivity in the pellet? We find radioactivity in the pellet because this viral DNA has entered into the bacterial cells, and bacterial cells have settled down in the pellet. That is why radioactivity is associated in the pellet with the bacterial cells, with the infected bacterial cells. Whereas no radioactivity was found in the supernatant, because in the supernatant we have exclusively ghosts, that is empty viral protein coats. So this indicates that DNA of this radioactive DNA of this T2-phage has entered into the E. coli. E. coli is heavier, it has settled down in the pellet, and that is why radioactivity is in the pellet, because of the fact that viral DNA of the T2-phage has entered into the bacterial cells. In the second experiment, in the second case, we have culture B, where we have culture B, and in this case T2-phage were taken, but instead of this DNA, they made the protein coat radioactive, and this we indicate by the small lines. These small lines indicate that it is protein coat is radioactive by means of sulfur 35. That means T2 were protein coat, radioactive protein coat. We have sulfur 35 in the protein coat. What is called as protein coat is also known as capsid. Then these T2-phage were used to infect the E. coli cells. They were used to infect E. coli cells. This is E. coli cells. And obviously what we find that this phage, it will enter its viral genome into the E. coli. So this is viral genome. So this is ghost empty viral protein coat, and this is viral genome. viral genome. Then blending. During blending in a blender, these bacterial cells were shaken or agitated in a blender so as to break the physical contact between the virus and the E. coli. This is E. coli. So what we find? We find this. This is viral genome. This is viral genome, and plus we have empty protein coat, radioactive empty protein coat, what is called as ghost or capsid, empty capsid. This is empty capsid, but radioactivity is associated with this capsid, with empty protein coat, with this capsid. This radioactivity associated with this capsid. Now centrifugation. We have explained infection, we have explained blending, we have explained centrifugation. We know that during centrifugation in a centrifuge tube, these heavier particles, they will settle down in the bottom, that is in the pellet. They will form the pellet, and the lighter particles, they will be in the supernatant. So we have here we have protein coat, the empty protein coat, what is called as capsid, empty capsid, and this radioactivity is present here. This is radioactivity is present here in the supernatant. Radioactivity in supernatant. So radioactivity was found in the supernatant. No radioactivity present in the pellet. While in the pellet we have bacterial cells. Infected bacterial cells. So bacterial cells in pellet. No radioactivity in pellet. No radioactivity in pellet. No radioactivity in pellet. So now what we find? We find that all the radioactivity was present in the supernatant and no radioactivity was found in the pellet. We know that in the pellet we have bacterial cells, and in the supernatant we have protein coat. This protein coat remains outside the bacterial cells. It means that this protein has not entered into the bacterial cells. If the protein would have entered into the bacterial cells, then this radioactivity would be have in the pellet. But protein does not enter into the bacterial cell. That is why this protein coat remains in the supernatant and radioactivity is found in the supernatant. No radioactivity in this case, no radioactivity is found in the pellet. This pellet contains pellet contains what? Infected bacterial cells. So this clearly shows that it is the DNA of the. So what this shows? It shows that it is the DNA of the T2 phage which has entered into the bacterial cells. It is a DNA of T2 phage entered into the bacterial cells, entered into the E. coli cells. So it means that it is the DNA which is the genetic material. Protein is not the genetic material. Actually, this in the earlier times, protein was having a suspicion that it may be a genetic material because of the diversity. Proteins are much more diverse as compared to that of the DNA. But then several evidences, several experiments proved that no, it is the DNA which acts as a genetic material. Protein does not act as a genetic material. So DNA acts as a genetic material in living organisms. So this DNA is a genetic material, and this contains genetic information. So it is a genetic material in living organisms. DNA is a genetic material. Genetic material. I told you that. So in this way we have given another evidence that it is the DNA of the phage which have entered into the bacterial cell. So DNA is the genetic material. These Hershey and Chase, they used the term blender experiment. They used the term blender experiment for this transduction experiment, for this bacteriophage experiment. Blender experiment, because of the fact that this phage, the virus and the E. coli are mixed together, they are blended together in the agarose. That is why they use the term blender experiment for transduction experiment. Now, what do you mean by transduction? What do you mean by transduction? What do you mean by transduction? Transduction is simply the transfer of genetic material by means of phages. That is called as transduction. When the phages get involved, when the viruses get involved in the transfer of the genetic material, we say that it is a transduction. So transduction is the transfer of genetic material by means of viruses. Viruses involved in viruses, that is phages, involved in transfer of genetic material. Transfer of genetic material. So transduction is the process where the genetic material DNA is transferred from one organism to another organism by means of phages, by means of viruses. So viruses, when viruses get involved in the transfer of genetic material, we say that this process is known as transduction. And another term we use it here, that is ghost. What is ghost? What is ghost? What is ghost? Ghost is simply the empty viral protein coat. So we have this. This is the protein coat. This is empty protein coat. This empty protein coat is known as ghost. The empty protein coat of phage, the empty protein coat of virus is known as ghost. So thanks.